GCSE revision video on required practical 2, and again the importance of this is that required practicals will count over 15% of the marks in your 2 GCSE biology exams. Practical is biology only, so if you do combined science, please do not watch this video because it won't be much value to you, and it is about microbiology. So some information you need to know, because you need to be able to link the content and the theory to the practical. That was Alexander Fleming who discovered antibiotics, and he discovered the first antibiotic, which was penicillin. You need to know that name. Why was it called penicillin? Because it was isolated from the penicillium mold, which he discovered. Many different stories on how he discovered it, but he discovered that it was killing bacteria that was growing on an agar plate. It also is important to know that antibiotics kill bacteria and cure bacteria diseases, but they are not a useful drug against viruses. They do not kill viruses. What would you be expected to know then? What you'd be expected to know is something like this. So you need to know that what Fleming saw was clear zones around antibiotics. So he obviously just had penicillium mold there, but the clear zone shows you where the bacteria doesn't grow due to the antibiotics. And what you did is replace that with paper soaked in antibiotics, and that gives you the clear zone around the outside. So the bigger the clear zone, the better the antibiotic at killing the bacteria. And you would have to apply the maths equation, area equals pi r squared. So what you would assume, unless they tell you in the exam, is that pi is 3.14, and obviously r squared is the radius squared. So how do we work out the radius? Well, some people measure the piece of paper outwards, but a strong suggestion to you is measure the diameter and then halve it. So the whole diameter of the circle. Some people make the mistake of taking the paper away, but there wouldn't be bacteria growing underneath that paper if there's a clear zone around the outside. So measure the whole diameter of the circle, half it, and then radius it. So if this diameter of this circle was 4 centimeters, you'd be doing 3.14 times 2 squared. Okay, so let's move on. Let's try and move on. You need to know, and how does this practical work? Or what you did, it's unlikely that you'll be probably asked to do this exact practical within a method, but what you would have done is clean and disinfect your hands and workspace. So the key word there is disinfect. So don't just say clean, you need to be using disinfectant to clean the workspace. You would then have wrote on your petri dish the different antibiotics you were going to do. So split your petri dish up with a pen and then put a dot where you're going to put each paper disc and label what the different discs have and put your name and date on that. You would then collect your discs, lift the lid from a minimum distance away from you, and we'll talk about why in a few moments. Place the paper onto the spot that you've drawn and do it again for the remaining discs. You would then put a small tape around the side of the petri dish, and then you'd incubate at 25 degrees C for 48 hours before measuring and recording the diameters of the clear zone, and then calculating a mean diameter of the area of the zone. So that practical, I don't think you're likely to be asked to write that as a method, but you could be, so it's worth making sure you could talk your way through it. Questions about the method that you need to be able to answer. So first of all, why do we disinfect the workspace in our hands? Well, that is to avoid contamination, because we don't want to actually add any bacteria to this that aren't on there. There's already bacteria growing on the agar. Those are the only bacteria we want to be on the agar. We don't want to introduce unwanted bacteria. We lift the lid at an angle to prevent the, I don't know why it's not come up, but to prevent the entry of pathogens from the air. We incubate at 37 degrees because we don't want to grow pathogenic bacteria. So we grow it at 25 degrees in school because that should only produce safe bacteria. We do not completely seal the lid because we need oxygen to be able to get in there so the bacteria can respire. We take perpendicular measurements because the inhibition zone is not always a perfect circle, so we can calculate a mean if you carry it from 2 different directions. And then you see the answers to previous ones. And we use a plain disc, so one of your paper discs will not have antibiotics on it, because it allows us as what we call a control, not a control variable, just a control, and that allows us to compare it to other discs to see is it the antibiotic that's killing the bacteria, not just the paper. And what are the variables that you need to know for this experiment? Well, if you're given a table, don't forget you can use that to work out what the variables are, because the independent variable should always be in the first column. So the independent variable is the type of antiseptic in this table, or the type of antibiotic, depending on which investigation exactly you've done. The dependent variable is always this bit here, so the diameter of the clear zone of bacteria. Okay, that's the thing you're measuring, and the control variables then are the things you're keeping the same. So the size of the paper disc would be a good example, the time in the incubator, so 48 hours, the temperature of the incubator, 25 degrees C, and maybe the concentration of agar that you've used or the ingredients in the agar in any different petri dishes that you have used. But I would suggest the top 3 are the ones that you would recommend. We need to talk our way through aseptic technique. So let's imagine you wanted to collect some bacteria, say off your mobile phone, and grow it to see how well that bacteria is growing and what bacteria is on your phone. You need to carry out what we call an aseptic technique, which is basically a technique used to keep the environment as clean as possible and stop contamination of yourself and contamination of the bacteria. So step 1, you would wash your hands. Okay, so washing your hands with soap, you would need to say with soap in the exam to say to kill bacteria. Washing your hands without soap would make it worse. You would then take the inoculating loop, which is the thing that you will swab the bacteria with and transfer it to the petri dish, but it's obviously been used before, so you would take that inoculating loop and you would put it maybe in ethanol first and then in a Bunsen burner flame to kill any bacteria already on it before you take your sample. Once you've taken your sample, you would lift the petri dish lid off, but only at an angle, and that angle would be to prevent any bacteria from the air entering the agar plate. You would then seal the agar plate, put your name and date on it, but you would only seal it around the edges with the tape. You wouldn't completely seal it all the way around, and that is to allow some oxygen to get in there so the bacteria can respire aerobically. You would then incubate it at 25 degrees. We use 25 degrees in schools because if we used higher temperatures than that, it would increase the risk of growing dangerous pathogens, and we do not want to do that for obvious reasons in school. And then you would get your petri dish back out, okay, and you would look at your results. So you need to be able to talk about risk assessments of this, so the fact that you could get exposed to bacteria and how you would minimize those risks and how you would prevent those risks as a result. So some questions that you need to be able to talk about with all these is what are the variables, and you need to be able to understand the maths and remember how to do the maths. So the independent variable in this experiment, type of antiseptic. The dependent variable is the diameter of the clear zone. The control variables are the size of the paper disc, the temperature incubated at. The equation for area, which you need to know for maths GCSE as well, is here. And you will also need to know which one has the largest effect. So it's the one with the largest clear zone. Don't make the mistake of getting radius and diameter muddled up. So my suggestion is you measure the diameter and halve it to get a better measurement. And obviously you will need a ruler in the exam.